So everything that we've talked about so far with oxidative respiration is basically the process by which a cell takes a molecule of glucose and utilizes the energy stored in the chemical bonds in glucose to make ATP. And by having oxygen present as the final electron acceptor, it turns out we can make quite a bit of ATP. But the question is, what happens if oxygen isn't present to be the final electron acceptor? Well, then we get this process called fermentation. And it turns out fermentation is the process by which we make things like cheese, wine, and bread, which are all yummy, good tasting things. But in terms of energy reward for effort, this is not a very high yield process. It's one that cells use only if they have to in the when oxygen is not present. So let's look at this process of fermentation. When oxygen is not present for a cell, that creates an issue for it in terms of generating ATP. It's got to find some other final electron acceptor for the electron transport chain so that it can continue to manufacture ATP, or it's going to have to go through some other route called that I'm going to talk about in a second, called fermentation. Now, there are some organisms out there, for example, some types of bacteria, that undergo a process called anaerobic respiration. And what that means is that rather than using oxygen as the final electron acceptor, these organisms utilize some other molecule as a final electron acceptor. Commonly, things like sulfur atoms or nitrogen atoms can be used as the final electron acceptor, but it's some sort of inorganic molecule that's used as the final electron acceptor. That's a relatively rare. Okay, not very many organisms use that pathway compared to how many use, for example, oxidative respiration. Another option is, and this is what yeast cells do and many types of bacteria do, they undergo a process called fermentation when oxygen levels plummet. When there isn't enough oxygen around, they go through an alternative strategy to find something else to accept the electrons at the end of all of their work of breaking apart chemical bonds. And in this case, they use some sort of an organic molecule as the final electron acceptor, but they do not use oxygen because oxygen isn't there. Now, there are different types of fermentation I'm going to talk about in just a minute. There's alcoholic fermentation, which is what the process that's used to make things like beers and wine. And then there's lactic acid fermentation, and that process is actually what occurs inside our own muscles when you go out and sprint on a track or whatever, and your muscles can't get enough oxygen to keep pace with the demands being made on them. We'll talk about that in just a minute. So again, fermentation means that I'm using some organic molecule as the final electron acceptor, and this occurs if oxygen is not readily available. So in these cells, we still have glucose molecules, and first things first, the yeast cell, for example, will still undergo glycolysis. We will still split apart that 6-carbon glucose molecule into 2 molecules of pyruvate. Here we go. Here's our pyruvate down here. And in the process of making that pyruvate, we still get our 2 molecules of ATP produced, but then we're stuck. We can't go beyond this point. We can't send things into the Krebs cycle, into the electron transport chain, because there's no oxygen at the end of those processes to accept the electrons that are being passed around. So now we got a problem. What do we do now? Well, one of the things that the cells will do is they will convert pyruvate, which is a 3-carbon molecule, into a different 3-carbon molecule called lactate. That's one option. Another option is they may convert their 3-carbon pyruvate into a 2-carbon molecule of ethanol, or what we call alcohol, and also release a little carbon dioxide. Now, this process is a process of reduction. We are reducing the pyruvate. And in the process of reducing the pyruvate, we make either lactate or we make alcohol and carbon dioxide. And why would we do that? Well, we would do that because by converting pyruvate into lactate, we can regenerate new molecules of NADH. I'm sorry, of NAD plus from NADH. And why do we care about doing that? Because we need to have fresh NAD plus available in order to create new molecules of pyruvate, and it's only by creating molecules of pyruvate that I at least get 2 ATP molecules. I got to get some energy out of the glucose that I'm shuttling into the cell. So rather than going on and making another 34 ATP as I would using the Krebs cycle and the electron transport chain, the best I can hope for when oxygen isn't present and I have to undergo fermentation as a cell is to produce 2 ATP by splitting glucose apart into pyruvate and then converting that pyruvate to lactate so that I get the NAD pluses needed to make more pyruvate. Now, what about the alcohol and carbon dioxide way? Well, I could convert pyruvate to alcohol and carbon dioxide instead of converting pyruvate into lactate. And yeast, for example, does exactly that. Instead of converting it into lactate, it converts it into alcohol and carbon dioxide. The alcohol, obviously, is what makes a drink have its alcoholic quality, such as beer and wine. The carbon dioxide is trapped in, for example, beer, and that's what gives beer its bubbly or foam-like appearance. Now, what are the pros and cons of these processes? Oh, let me back up one more second here. I'm sorry, I forgot to mention. In we as humans, if you go out and sprint and run a 100-yard dash, for example, a 100-meter run, there's not enough oxygen stored in your muscle tissue to support that entire sprint, and there is not sufficient time for your circulatory system, your heart and blood vessels, to ramp up to speed with delivering additional oxygen to your muscles. So during that sprint, there will come a point when there's no more oxygen left in the muscle cells, and oxygen is not yet on its way to the muscles from the lungs, so that those muscle cells are in a state of where they have to undergo fermentation to continue making ATP to cause muscles to contract. And the way they do it is by this fermentation process here, where they're taking the pyruvate and they're just making lactate, or what's commonly known as lactic acid. And that lactic acid will begin to build up both in the blood and in the muscle tissue, and we believe that's what contributes to things like muscle aches the day after sprinting or working out under anaerobic conditions. It may also be what causes athletes to fatigue and not be able to finish as fast as they started. So what are the pros and cons of fermentation? Well, probably the single only advantage to it is it does allow cells to make molecules of ATP even when oxygen is not present. That's about the only advantage. There are big drawbacks to it, though. You only get 2 ATP total that you make out of breaking apart a glucose molecule. That's a very low amount of ATP produced. Compare that with oxidative respiration that we already learned about, and you know there we make somewhere on the order of 36 to 38 ATP molecules from 1 molecule of glucose. That's a big difference. So fermentation is not a process that a cell would typically undergo unless it had to. There's just not enough of an energy payback to make it worth the cell's while unless it has no other options available to it. Now, the last thing I want to talk about is the idea that we can get energy from molecules other than carbohydrates. Our cells are pretty much primed to convert sugar, glucose, into usable energy in the form of ATP. But our bodies can also take other types of organic molecules and harvest energy from them in order to manufacture ATP. So we can take proteins and break them down into their individual amino acids and alter them in such a way so that we can produce acetyl groups that can then enter into the citric acid cycle. We can also take fats and through a process called beta oxidation, we can convert fatty acids and glycerol into molecules of acetyl-CoA, and once we have acetyl-CoA, as you know, you can readily enter into the citric acid cycle and make ATP, and then make additional ATP in the electron transport chain. So while carbohydrates are the preferred form of molecule we use for getting energy, to make ATP. They are not the only means by which we can do that. We can rely on other organic molecules such as proteins and fats to get us energy. These the cumulative caloric reservoir that we have between proteins, carbohydrates, and fats is called the metabolic pool. And finally, just to remind you one more time again here, we've got a couple of different terms that relate to metabolism that are involved in taking these organic molecules in the metabolic pool and converting them into ATP. When we are breaking down proteins, carbohydrates, or fats and using them to generate ATP molecules, we are undergoing catabolism. That is the breakdown of molecules into smaller parts and the release of energy. In the case of amino acids, this event occurs in the liver and it's called deamination. In the case of fats, this also occurs in the liver and is called beta oxidation. You will remember the opposite of catabolism is anabolism, and this is where we take smaller building block molecules and we join them together to make a larger molecule. For example, if we took a bunch of individual amino acids and joined them together to make a polypeptide chain.